The subject of our lesson at the moment is a discussion of how it is that my adaptive lymphocytes develop the receptors that they have on their surface. Remember, I differentiated in previous discussions the difference between innate lymphocytes, which do not have this same style of receptor, and then my classically known lymphocytes, my B and T cells, who have receptors on their surface which uniquely bind to three-dimensional shapes of molecules that they find to be foreign. So we call these the B cell receptor, or BCR, or the T cell receptor, or TCR. And if you notice, these two molecules are encoded in the same superfamily of genes. We refer to this as the immunoglobulin superfamily, and it's a group of related genes which create membrane surface receptors that have something to do with cell activation or communication or signaling in some way. Now, when you compare the two receptors on the B cell and the T cell, you'll notice that they have very many similarities, but a few differences. They each have an N-terminal end of sequences of amino acids that will be outermost off of the surface of the cell, and a carboxy terminus, which is going to be important for transducing the signal through the cell membrane when something has bound to the N-terminal end. Now, in a later discussion, we'll actually talk about how these N-terminal ends are derived uniquely, but for now, let's just look at the big picture of how the two receptors are similar and different. I've drawn them side by side here so that you could see some comparisons. Notice, as I said, that they are both basically Y-shaped structures. The BCR on the left and the TCR on the right are both basically shaped like a Y, the difference being their composition, that my B cell receptor is made up of two identical long chains and two identical shorter chains, and we refer to those respectively as heavy and light, referring to their molecular weight. The heavy chain has more amino acids in it, the light chain has relatively fewer. But the fact that there are two copies of each of those chains on either side of the molecule held together by a hinge region means that it's going to be possible for this molecule to have some flexibility in the distance between the things it attaches to. Now, if both of the chains are identical, I think that you should be able to imagine that the three-dimensional shape of the N-terminal groove that forms on each side of the molecule must be identical. Because if I have the heavy and the light on each side being identical to the other side, there's no way to make a different shape. So I think the easiest way to think of this is to imagine yourself with your arms outstretched and your two hands being the three-dimensional pockets into which a foreign substance will fit. So you know that when you stretch out your arms, you are able to move your arms slightly closer or further apart, depending on what you are trying to grab onto. Well, an antibody molecule has some flexibility in that way as well. And I should mention, I just called this an antibody molecule. Realize that the freely secreted form of this membrane receptor that we're talking about now is just the same molecule, but packaged by ubiquitination for secretion instead of membrane insertion. So the B cell receptor is the precursor of all of the antibodies that we find free in our serum. Now, other than that, noticing that the N-terminal end of the molecule has the unique three-dimensional shape that would basically be the grip of your hand, realize that the medical terminology you need to know for that is idiotype. Idiotype is a word that signifies the antigenic specificity of a molecule. In other words, what precise shape does that groove have? Will it bind, for example, my stylus for my computer? Would it bind a larger pen? Would it bind the mouse for my computer? Each of those would take a slightly different shape. And the shape and antigenic specificity are determined by the idiotype, the N-terminal domains of both of the heavy and the light chains. Now, just to look across toward the shape of the T cell receptor on the side, realize that the T cell receptor has the similarity of shape, and yet I have only one of each of the types of chains that are going to make it up. And because of the fact that those two chains are similar in their molecular weight, I don't refer to them as heavy and light. I refer to them as alpha and beta. Now, that's simply a convention that immunologists and other medical scientists got into way back in history. If you could not designate differences between protein chains on the basis of size or length or weight, then you simply took the approach of naming them in the order they were discovered using the Greek alphabet. So alpha was the first one discovered here, beta was the second. Now, because I said to you at the beginning that these are products of a unified superfamily of genes, you should realize that there are analogies in the function of these two sets of molecules. And the beta chain on the T cell receptor is actually going to act quite similarly in our next module when we start talking about rearrangements of the genes that make the cell's receptors unique. So right now, I will repeat this again in a later module, but if you think about the word big and you spell it with a beta at the beginning, instead of a English B, think about the fact that that is almost a synonym of the English word heavy. If something is big, it's usually heavy, right? So what I'm saying to you is that the heavy chain of a B cell is going to be somewhat analogous to the beta chain on my T cell. And that will come to light later when we start talking about the complex genetic rearrangements that we will do in the germline in order to make the unique molecules for each cell. Well, if beta on the T cell is equivalent to heavy on the B cell, then it stands to reason, I think, that by process of elimination, you can assume that the alpha of the T cell is going to do something similar to the light chain on the B cell. So this is not something you will need right now, but analogies between these will become useful in a short period of time. Now, if you notice, other than that, we have some flexibility built into the B cell receptor through the hinge, which allows the two arms to stretch either closer or further apart from one another. We then have a group of what are called constant domains that are downstream from that N-terminal end. That should imply to you that unlike the extremely variable domains that made up the N-terminal end and were unique to the idiotype of the molecule, the constant domains are going to be much more similar in amino acid concentration to one another. And they are what are going to give the molecule its isotype or family. Okay, you probably already know the isotypes of B cell receptors are defined by the constant domains of heavy and light. So when we talk about those, we again revert back to the Greek alphabet. If a heavy chain is of a mu type, Those would be the two put together heavy chains that would make up an IgM molecule. Delta would make up IgD, etc., as we go down the list. So we simply took the Greek letter that pertains to the English letter for that type of immunoglobulin or constant domain. Now realize that those names also designate what I just termed the isotype of the molecule. So the family is IgM or IgG or IgA. The family is also the isotype. Now, similarly, my light chains have isotypes as well, and I refer to them as kappa and lambda. We sort of ran out of order in the Greek alphabet and simply picked two letters that were easy to write. Now, the isotypes in the T cell receptor are just named according to the chain because it's only the difference between N-terminus and carboxy terminus, and so we simply call them alpha beta isotype, and there is another much more rare isotype that we really do not fully understand what it does, that is called a gamma delta T cell. Okay, those cells actually predominate in the gastrointestinal mucosa and are prominent on the surface of several types of innate lymphocytes, so they clearly have a different role, perhaps in evaluating things that pass through your gastrointestinal tract. Is this food that I should obtain nourishment from, or is this indeed some sort of invader that I need to respond to immunologically? But as I said, they really can't ask you about that other isotype of T cells very much because we know very little about it. All right. Now, other than that, let's see what I didn't get to on the diagram. We talked about isotype. We talked about idiotype. We haven't yet talked about allotype. Allotypic markers are simply very minor amino acid concentration differences that are found within the constant domains of the chains of the receptors. So I can have allotypes of either type of receptor, and remember that these are genetically inherited from your parents. So it's technically possible that you could analyze these allotypes in cases of questions of paternity, and you could rule out an individual who could not possibly be the father of a given child. But I have to say to you that that really is a cumbersome technique that is not used anymore, and we now have much more refined and precise ways of doing it that don't depend on the immunoglobulin allotype. But technically, it's simply an inherited difference in amino acid concentration. It does not affect the way the immunoglobulin or the receptor works. It's just sort of a fingerprint that you inherited from your parents. Now, notice that the carboxy end of each of these molecules extends through the membrane when it's functioning as a receptor. And so it's going to be the case that when something exciting goes on in those N-terminal end pockets, reflecting that something foreign has been bound, there's going to be a shiver of excitement sent through that membrane that will pass through what we call a signal transduction complex, and that will result in a cascade of events that are going to be phosphorylation pathways that will both activate and cause the cell to clone itself. So think about the binding to a foreign substance being that first step that's going to cause my cell to wake up from its sleep as it's wandering around the body and think to itself, aha, there's something going on, I must make a response, and I'm now going to make copies of myself so that not only will one of me be available to make a response, but there will be a growing family able to do that. And that, of course, is the way we develop our immunological muscles. The signal transduction complex in T cells is referred to as CD3. The signal transduction complex in B cells is composed of CD19 and CD21. And another thing that we can call that, and you may see this terminology, is that they may refer to this as being the pan-T or pan-B cell marker. The idea being that all T cells or B cells would have one or the other of those, so it becomes an effective way to count these in clinical practice. Okay? Now, beyond that, if I were to compare side by side the B cell receptor and the T cell receptor, in summary here, notice that the distinction is that I've got two copies of the two chains in the case of the B cell receptor, whereas in the case of the T cell receptor, I have only one copy, and they're different in molecular weight. Okay, very heavy versus light in the B cell, equivalent in molecular weight, and therefore alpha beta in the T cell. Now, as far as function is concerned, notice that the B cell receptor will bind to any chemical composition. There are B cells in your body that will recognize lipid. There are some that will recognize nucleic acids. They may recognize polysaccharides. There's no limit to what potentially could fit that three-dimensional groove on the end of the arm. But notice that it's an absolutely tremendously important fact for you to understand for the T cell, that T lymphocytes can only recognize peptides, small chunks of proteins that are being presented to them inside the groove of another molecule known as the major histocompatibility complex, or in humans, the HLA. HLA stands for human leukocyte antigens. MHC stands for major histocompatibility. Those are the same thing. It's just that we call it HLA in the human. MHC is the general term for that sequence in all creatures. Okay, now next we have the fact that each of these receptors must only be present in the form of one type per cell. Because remember, the entire specificity of my immune response is determined by one cell recognizing one unique thing and making a unique response against it. So I can only have one antigenic specificity per cell, although notice I can have two copies in the case of the B cell receptor, because it has two hands on the end of two arms, right? So two copies that recognize the identical thing. The T cell recognizes only one, fitting in the groove in between the alpha and the beta chains. The isotypes per cell, when we're talking about the receptor, B cells are capable of having either one or two, depending on their time of development. You probably remember that mature but naive B cells coming out of the bone marrow will have two. They will have molecules of M and D on their surface when they come out of the bone marrow. But when they are mature and are capable of responding, they will switch over to having different isotypes of IgG or A or E as their membrane receptor, and they will no longer express D by the side. That's going to be the topic of another module, so I'm just introducing things now. Remember, the B cell is slightly flexible in its receptor through the hinge. The T cell is not. It's designed to recognize only peptides being presented in another rigid molecule. So it has to have a complementarity for one type of structure, and it simply does not wobble at all in its ability to recognize. The signal transduction complexes, CD3 in the case of the T cell, CD19 and 21 in the case of the B cell. And remember that another key thing to remember is that B cells receptors are truly designed eventually to be secreted and released free into the plasma. Now, the cell that actually does that, if you recall, is the plasma cell. So it will do the secreting into the plasma. But it is the membrane receptor in a slightly different format that is actually predestined to become your serum antibody. And the T cell receptor is never, ever released from the membrane of a T cell. It has no role independently by itself. Now, I've introduced a new idea into this particular module because I've been getting feedback from students on the forum that they sort of enjoy being challenged in this way at the end of a particular topic. And so this is going to be my hashtag MMC module, which means, abbreviated, make me care. Because it's very easy when I'm drawing things like this and talking to you about idiotypes and allotypes and isotypes, and you're staring at your computer, even if you're doing it at super speed, you're still glazing over a little and wondering why on earth a practicing physician would need to understand this. So let's ask a couple challenging questions here. And what I'm going to do is I'm going to pose the question, and then I'm going to suggest to you that you shut off the audio for a moment or pause the presentation and then come back when you've thought through the concept yourself. Because what would be really nice is if I present the question, give you time to think about it, and then when I tell you what my answer is, your answer and my answer agree with one another. So use the pause button here and try to think through this on your own, because what I'm trying to do here is sort of challenge you with where are these things important in medicine. So first of all, how do you suppose, out of the molecules that we've talked about so far, the B cell receptor, the T cell receptor, and signal transduction molecules, which do you suppose would be the molecule that would be best in clinical medicine to count populations of B cells or T cells? So pause and think for a moment. I'm going to go ahead now as soon as you unpause it and explain. Well, if I go back to my table in summary, notice that each one of these structures is absolutely unique to a single B cell or T cell. The receptors that they each have are unique. So I'm not going to be able to use the B cell receptor or the T cell receptor itself to count entire populations of B cells or T cells. What I'm going to want to use in that case is my signal transduction molecule, because I know that every single B cell in your body has to have CD19 and 21, and every single T cell in your body has to have CD3. That means that if I count that number by flow cytometry, I'm going to get a count of the total number of cells of that type that you had in a volume of blood or serum or whatever. Okay, next MMC. Epstein-Barr virus infects cells that possess CD21 on their surface. And the cells that have that molecule, you already know B cells, but also we find CD21 on oropharyngeal epithelial cells. So what type of malignancies do you anticipate that I would have in a patient if neoplasia results as a sequela of Epstein-Barr virus infection? So now pause the video for a second for yourself as you think through this. And the answer you should come up with then is going to be B cell lymphomas and nasopharyngeal carcinomas. Now, if that was a little too much into path or micro for you, just remember there aren't any subjects anymore, there are only patients. So thinking about what the sequela of infection of a cell that bears CD21 is going to have huge significance to you in clinical practice. Okay, next. Patients with multiple myeloma and Waldenstrom's macroglobulinemia have neoplastic plasma cells. Remember, my plasma cells make antibody. So what do you think you're going to find in the urine of these patients? Test yourself by pausing the video now, and I will now say that they are making huge amounts of antibody. That's what plasma cells do, is just overabundantly produce antibody. And so when that reaches the kidney is going to be able to get the protein molecules out that are very large, so it will handle the heavy chains, but the light chains in overabundance end up being too much, and they end up being free in the urine. And if you recall, we call these Bence-Jones proteins, and they are a signature, basically, of a person who is making way too much antibody because of the malignancy of the plasma cells, and because it overwhelms the kidney's ability to filter, these very small light chains end up in overabundance in the urine. Now, interesting side of this, in the human, and we cannot explain why, twice as many immunoglobulin molecules use kappa light chains as use lambda. Again, we don't know why that is, but in the human, most Bence-Jones proteins end up being kappa light chains. There's a 50% less chance that they would be lambda light chains. And that's just sort of an oh my that we couldn't ask you much about because we can't explain why that's true in the human. Okay, next Make Me Care and final one for the module. This is getting way ahead intellectually, but could you predict why the immune response to materials that contain protein is going to be significantly different than the immune response to things that don't have protein, things that are purely lipid, purely polysaccharide, purely some other type of molecule that contains no protein? Pause the video and think about this for yourself. And this one might be a long pause for you because it really gets at the crux of how the immune response works. But let's go back to looking at what T cells and B cells see. Notice that out of these two cells, the T lymphocyte can only see protein. And remember, I characterized in my first module that my T lymphocyte is really the brains of the operation, the general of the army who is going to transmit the signals. So if he or she, the general, can only see peptides, then it means that any orders that are coming toward other cells are not going to be possible unless there are protein materials in that antigen. This is a very important premise for all of immunology, and if you didn't totally wrap your mind around this now, remember that we will repeat that principle over and over and over again so that you can understand how important it is for all of the signaling of your immune response to be delivered from your general to others. With no protein in the material, there's no signaling from the general, and therefore any immune response that I did create would be much diminished in size and speed and effectiveness. Well, thank you for listening, and I'll see you next time.